Perfect. So, welcome everybody to the Geobiology and Geomicrobiology seminar series at Bristol University. It's an effort between the School of Geographical Sciences and Earth Sciences. The host today, my name is Patricia Sanchez-Baracaldo, and my co-host is Bryce from the Earth Sciences Department. In terms of announcements, I was going to say that we're starting to record the seminar series. They will become available at some point, but we're sort of organizing that at the moment. And for me, it's a real pleasure to introduce today Paula Welander. We met, I think, a couple of years ago in the astrobiology conference in where was that? I was in Atlanta, and Paula is originally from LA. That's where she did her undergraduate in Occidental College. Then she did a master's and a PhD at Illinois University in microbiology, and then she did her postdoc at MIT with Diane Newman, Roger Summers, and you also work with Tanja Bosak, I believe, and that's when you got introduced into the geobiology. So yeah, please, go ahead and get started with the talk. And
great, thank you.
By the way, thank you for saying agreeing to the talk today. So over to you, Paula.
Thank you for having me. So let me get this here. It's great to see so many people on the call. This is amazing. I'm so excited to share this with you. Hang on a second. Okay, there we go. All right, well, great. Thank you all for coming, and thank you, Patricia, and let me just change this to, oops, so I can see myself, for inviting me, and Casey for inviting me, and for that lovely introduction. I'm really excited to share with you the work that's been coming out of my lab. Some of this work has even come out during COVID time, so my students are really working hard to keep us going and keep our group and research going and stay sane in these crazy times. So I'm going to be talking about some work that we've been doing to try to test the sponge biomarker hypothesis, which I will describe in a bit. But before I do that, I wanted to kind of step back and give you a little broader kind of interpretation or idea of what my lab works on and how a microbiologist like myself fits into geobiology. So geobiology is a very broad field which encompasses a lot of different scientists into a lot of different fields. And if I have to think of one theme that kind of goes through geobiology, is that we're all very interested in trying to understand how the evolution of the Earth over time and the evolution of life have impacted each other, how these two processes have pushed and pulled each other throughout time. And so if you're a geobiologist who's interested in this and how these two different processes have impacted each other, say when the Earth looked like this, then the type of life you're interested is microbial life, because for the majority of life's existence on this planet, life has been microbial. And so that raises an interesting questions if you're trying to link these studies to the rock record, and that is, how do we study microbes in the context of Earth's distant past? How do we study them and link them to the geologic record? And so geobiologists that like to study this actually are quite creative, and there are a lot of ways that we can link microbes to the rock record. And one of these ways is to actually use morphological structures that are preserved in the rock record. And these include things like actual cells that could be preserved, or actual bigger structures that are representative of microbial communities, like these stromatolites shown here. So just like we find dinosaur bones in the rocks, we can find morphological signatures of microbes. We also can do chemical signatures. And so we can, microbial metabolisms can leave certain isotopic imprints in the rock record that can then be deciphered and pulled out by geochemists. And microbes can also actually leave actual chemical fossils, actual molecules that they deposit and are preserved, and we can use as biomarkers. And so that's what my group focuses on. We've been focused on these molecules shown here and using these as potential fossils. So how does that work? So the lipids as molecular fossils is a very straightforward process. For example, you have this molecule shown here that's produced by some ancient microbial community. These microbes die and settle into the sediment, and the majority of what the microbes are made of, with DNA and protein, that all gets degraded. But molecules such as these are actually quite recalcitrant and can be, in the process of the sediment becoming a rock, can be fossilized into this type of phospholipid. We lose a lot of like the hydroxylations and the other kind of bells and whistles that are present here, but the basic chemical structure can be preserved. And then we have geochemists that are able to go into these rocks and extract these molecules and are able to detect them. And then based on the ratios of certain molecules, and based on what organisms we know today produce these molecules, we can make certain interpretations. We can make interpretations about what taxonic groups may have been present at the time of deposition, what metabolisms may have evolved at that time, and even perhaps what the environmental conditions were like at the time of deposition. So there's a lot of different steps to using these molecules as fossils, right? Everything from taphonomy, how are they preserved, to actually finding them in the rocks and detecting them. And we do none of that. My group doesn't focus on anything. We don't leave a lab. We don't do any field work. We don't touch rocks. What we work on is how can we use our ability to study microbes in the lab to better enhance the interpretations that are made of these biomarkers. What can we contribute in our understanding of how these molecules are made and why these organisms are making it under what conditions, and can that help us better constrain a lot of these interpretations that still remain an open question. And so we use these molecular tools. We thus far have used a very kind of common way to do this in my lab. And basically what we'll do is we'll identify a molecule that we're interested, that some question needs to be answered, or an organism that makes some molecule, but we're not sure exactly how it makes it. And so the first question we ask is, how does this microbe make this molecule? And what I mean by that is, what is the biosynthetic pathway? What are the actual proteins that are involved in the process of converting some precursor molecule into the final lipid that is eventually utilized by the cell and then also preserved in the rock record? If we don't know that, we figure that out. And we've been working on a lot of different biomarkers to try to figure out exactly how different microbes potentially make these. Once we identify the biosynthetic pathway, that provides us a whole wealth of tools we can use to better constrain these biomarkers. One of the things that we like to do is we can study the biochemistry of this. We can actually look at the structure of the proteins and determine the biochemical mechanism for how protein X and Y convert this molecule into that molecule. And we can identify differences in different organisms. Sometimes we find that eukaryotic organisms have a different protein for doing something that, for making the exact same molecule that microbes are making. And so those differences we could potentially then identify through isotopic analysis in the rock record. We can do physiological studies. So we can make mutants where we block the production of this by deleting these proteins. And then you'll have your wild type strain, which produces the lipid, and some mutant that doesn't. And we can test how in the lab that affects its growth under different conditions that are relevant for the environments we usually find these microbes, right? So we can start to link environmental conditions and physiological growth and response to the presence of these lipids. And we can also do a lot of bioinformatic work. So once we know what the proteins are to make these, we can look in our genomic databases, which are very large at this point in terms of microbial organisms, and identify other organisms that could potentially be producing these lipids, right? So typically we find what organisms make these lipids by doing lipid analysis, but using genetics and these genomic databases, we can actually increase or even constrain further the organisms that could produce this. We can also do molecular clock analyses to determine when certain groups evolved these pathways and kind of try to date that with the geologic record. And we can also do try to trace the evolutionary history of the biosynthetic pathways, which organisms actually evolved these first through these types of bioinformatics analyses. So that's kind of the approach that we take. And so in my group, I have several different wonderful scientists working on a variety of different lipids and biomarkers and questions that we're trying to answer. And each one of these wonderful people are working on a different aspect of these different types of biomarkers. And so today we're going to focus on Mallory's work, which she has been doing on trying to better constrain these biomarkers that are being used as sponge biomarkers. So these are sterol molecules, and sterols are one of my most favorite molecules to study. These are these tetracyclic compounds shown here. And this is cholesterol, which you recognize probably as what us and many other vertebrates produce. And sterols, as a biologist, to me are very exciting because they have a lot of different functions. I think the most well-known function that sterols have are this membrane structure and fluidity. They are known to provide structure when our membranes become too fluid under different conditions. And so they just have this ability to really kind of reinforce our membranes. But they also have a lot of other functions, particularly in more complex organisms. You can have them be precursor molecules to steroids. In certain yeast, they can act as oxygen sensors, so they will allow the organisms to sense when they're running low on oxygen and switch their transcriptional profile. They're also insignificant in pathogen interactions. So there are a lot of intracellular organisms that infect human cells that actually use cholesterol as a source for carbon. And Mycobacterium tuberculosis is an excellent example of this. So these molecules have a very important role in all life, and it's why they're essential for eukaryotic life. They're also very well preserved because of this tetracyclic structure. They function as biomarkers for very broadly for eukaryotes. And the reason they function very broadly as biomarkers for eukaryotes is because all eukaryotes require sterols. Even things like insects don't make their sterols, but they have to get their sterols through their diet. And usually by eating other organisms, they're able to get sterols. They're also used as biomarkers for oxic conditions because oxygen is required to produce these molecules. So if you see the sterins in the rock record, there was oxygen present at the time, enough to make these sterol molecules. Excuse me. And they're also well preserved in sedimentary rocks as far back as 1.6 billion years, which I think are the oldest biomarkers thus far. Okay. Now, so this is just the sterine molecule shown here that can be a very genetic marker for eukaryotes. There are also some sterols that are more diagnostic because they're more complex. And the reason they're more complex is because they have these kinds of modifications that I've shown here in red, which is methylations, ethylations, and propylations. And these are preserved in the rock record. And we have a pattern of these being produced in certain organisms today. And so they can be more diagnostic. So this brings us to the biomarker we're going to be focusing on today, and this is 24-isopropylcholesterol, or 24-IPC. In 2009, a paper came out that from Roger Summons' group by Gordon Love, and in this paper they identified 24-IPC as far back as 645 million years ago in the Cryoxenian period. And because sponges at the time were thought to be the predominant source of this molecule, this indicated that you could see, a record of sponges into this period. But this was a little contentious because if you look at the morphological record of sponges, that pushes it back. I've learned now that some of these morphological fossils are also controversial. But depending on which ones you think are the real sponge morphological fossils, they could push the record back of sponges, over 100 million years ago to 45 million years further back than the actual fossil record that we're seeing here. And so there's this gap, right? And so, which is exciting and great, but it is also controversial. And so there's been a lot of back and forth in the literature about whether these are actually good biomarkers for fossils. And there's a couple of reasons why this argument is made. One of the reasons this is thought that you could question this as a biomarker for sponges is that there are, like many other biomarkers that we use, alternative sources to these biomarkers. And so here I'm showing you that there's papers showing that pelagophyte algae can produce this N-propylcholesterol and make some amount of isopropylcholesterol as well as these proteins, these Rhizaria protists, are also known to produce primarily N-propylcholesterol, but also produce some of this isopropylcholesterol, right? And so there's things like this where you have this discrepancy that kind of question whether this is actually the appropriate biomarker if we're using it appropriately. So being that we are interested in the biochemistry of these in the biosynthetic pathway, the first question we ask, well, what do we know about the synthesis of isopropylcholesterol? So it turns out that methylating at the C24 position has actually been studied in some organisms. And so this occurs through this enzyme known as the C24 sterol methyltransferase, or SMT. And it's been shown in yeast that they can methylate at the C24 position once, and they've identified an SMT that is able to catalyze that reaction. And in plants, we know produce ethylated sterols. And in plants, it's been shown that they have 2 sterol methyltransferases. And we have yet to find a sterol methyltransferase that is actually able to propylate at the C24 position. And so the question that we have is, well, what do we know about these steromethyltransferases and these other organisms that we do know produce isopropylated sterols, and has anything been done? In 2016, David Gold, who was a postdoc at the time in Roger Simon's lab and is now a professor at UC Davis, looked into this and looked into sponge and other eukaryotic genomes to see if understanding the genomic context of these could help provide some insight and some clarity into these other organisms or these other alternative sources of isopropyl cholesterol. And what he found was a very interesting pattern. He found that the number of SMTs in a eukaryotic genome actually correlated with the number of alkylations. So if you had, well, no SMT, you're not going to methylate, right? But if you have one, you methylate once, two, three, so forth and so on. But this was an exception in sponges. What he found is in sponges is that you had one less SMT than the number of methylations that you were seeing. So if a sponge was shown to ethylate its sterols, it had only one SMT instead of two. And if it had three methyl groups added, it had two SMTs. And so what David proposed was that there were two different pathways here to be able to get from two different sources to get to propylated sterols. In the algae, you have three SMTs that are required, whereas in the sponges, you have two SMTs. And so if you look down here, this final step then is carried out by two different enzymes. And he did a molecular clock analysis of these enzymes. And based on the relative, he was able to show that these sponge SMTs evolved before the algal SMT that would propylate. And so this was used as evidence that potentially, you know, these fossils that we're seeing here had to come from sponges because the algae SMT hadn't evolved yet, okay? And this is a wonderful analysis, and I think it's pretty robust. The problem is we have no experimental evidence that has confirmed this function here. And so we felt that this argument would be made much stronger if we could demonstrate that this is the pathway that we see. We see two different pathways for these organisms. And so this is where Mallory Brown, my student, picked up the project. And we had very basic questions. We wanted to develop a system in the lab where we could actually test these sterile methyltransferases. And the questions that we had was, you know, do these sponge sterile methyltransferase actually alkylate SE24? We see them in the genome, but no one has ever actually shown that that is their function. So from a very basic level, we just wanted to make sure that you can actually, that these enzymes are actually doing this reaction. And then, can these sponge SMTs carry out multiple rounds of reaction? If we have one SMT, will it ethylate instead of just like the yeast SMTs and just methylate, right? And then which SMTs are actually required to make propylated sterols? If a sponge has more than one SMT, which is the one, the algae have three, which one is the one that is actually propylating? So those were the questions we had. And so Mallory developed the system where, you know, she has all these sterile methyltransferases on the computer. We have them synthesized for us, and she's able to clone them into a plasmid shown here. She takes this plasmid and puts it in E. coli. We then lyse the E. coli cells, and she pulls out the proteins, all the different proteins in the cell. So she has a cell-free extract, which is just made up of a protein, all the proteins in E. coli, including the SMT that she's been able to express. And then she asked very simple questions. She incubated this sterile methyltransferase with two substrates, either desmosterol, which has no methyl group at the C24 position, or a 24-methylene cholesterol, which has a 1-methyl group at the 24 position. Okay, and then we tested for whether there was any alkylation at the C24 position. So we first looked at several different sponge SMTs, and what I'm showing here are the chromatograms with desmosterol. So these are no C24. This is our no SMT control. So this is what it looks like without any protein in there. She added the protein for several different sponges. I'm only showing 3. And sure enough, we see methylation occurring. So you can see in each one of these, you see this methylene peak appearing. And in some of these, she could actually see an ethylene peak appearing as well. And it wasn't a lot, but it was there. And it's definitely showing that these sponge SMTs are, one, functional, but they also potentially could be adding that second methyl group. And so we were wondering, this one should be able to produce ethylated sterols given its lipid profile. So Mallory decided to test with this substrate to see if we just needed to, you know, there could be substrate competition here. So if we gave it this substrate, we would see ethylation. And when we do that, we do actually see ethylation here. It's not a lot, and we're still working out on the conditions of this, but what it's showing you is that even in these where that all of these proteins are able to add both at a C24, whether or not there is a methyl group there, okay? So this is really promising, and Mallory went ahead and tested a whole bunch of different sponge SMTs, and what you see here is that all of them are able to at least methylate. And some of them in her first sample where there's no C24, she can even get ethylation to occur. She did have two where she saw methylation, but we did not see any ethylation. So, you know, we're still, you know, wondering if those are actually what we call promiscuous, where they can continue to add methyl groups or not. But overall, the work that she's done so far has answered some of our most basic questions, and that is, are these sponge SMTs actually sterile SMTs? And they are. So that's really great. So we have a system where we can show that. Can they carry out multiple rounds of alkylation? Yes, they can. And so, but not all of them. And we're not sure if that's a result of these actually not being promiscuous SMTs or just our system, which we're still working on. And then the one question we have is, which of these SMTs are required to make propylated sterols? Because that's the goal, right? We want to get propylated sterols in the tube. And so that's part of our future work. What we really want to test is multiple SMTs here. We want to be able to put in 2 SMTs from a sponge and see if we can make an isopropyl. The problem is that we don't have that data. It's very hard for us to find a sponge that makes isopropyl sterols whose genome has been sequenced, and we can get the proteins. And so if you saw that listed I showed you before, all of the ones that we looked at have one just SMT in their genome. So this is one of the problems with this project, is that, and David ran into this problem too, is the lack of data in terms of genome sequences available to us. Mallory's also working on testing the algal and rhizaria SMT. So what we're trying to do is move the system towards our experimental system to where we can have multiple SMTs, so we can actually show that we can make propylated sterols. Okay, so this is ongoing, but we also had something else we wanted to test. So we're wondering, so this is the hypothesis right now that is present with the eukaryotic SMTs that we observe in the eukaryotic genomes. And so we were wondering if there was potentially another pathway that could be out there. Is it possible that you have sponge SMT 1 and 2 that are just ethylating, and that there's a third SMT that's being contributed from another source that could lead to this? And the reason we thought about this is because sponges are very well known for having a very robust symbiotic community of microbes inside, right? Basically, a lot of us think that sponges are bags of microbes. And you can see here from this figure here, it's using 16S tags to look at the different types of microbes that are within the microbiome of a sponge. And the different colors are just the diversity of microbes that are present there, which you can see here on this pie chart encompass a lot of different phyla. And so on an unrelated project, I had been looking at these sterol cyclase proteins and biosynthesis proteins in a variety of environmental metagenomes. And this was several years ago, this popped up, which looks like a sterol synthesis pathway within the bacterial symbiote community of a sponge, Aplycina aerophoba. And what really struck me about this was that there were, in blue I've highlighted here, these sterol methyltransferases. And so this got me thinking, could potentially this be a contributor? I don't know if it's here or maybe further up, or is it even methylating these sterols? And so Mallory has done some further phylogenetic analysis of these sterol methyltransferases since the last time I did this. And she has actually found that there are a group of sterol methyltransferases that seem to be clustering together that we think of are of bacterial origin. And if we zoom in on this a little bit more, you can see that, yeah, here's the Aplycina aerophobia symbionts, but you also see that there are symbionts from other sponges, Patricia ficiformis and Thionella. But we also see that there are, from metagenomes from free-living bacteria in environments such as the Arctic Ocean and marine sediments that have sterol methyltransferases that seem to be of a bacterial source. And so this was striking to us because at this point, no one has even suggested that bacteria have the biochemical capability to methylate sterols at the C24 position. And so we had a very basic question. Are these actually methylating sterols at the C24 position, or are they doing some kind of different function? And so Mallory took her system that she had developed for the sponge SMTs and went ahead and cloned in her bacterial SMTs that she had found in these metagenomes. And we tested them. And so I'm going to show you a couple of chromatograms from some of the sponge symbionts that we were able to test. So this first set is from Patricia ficoformis is the sponge. We know it adds 3 methyl groups. Yeah, it propylates at the C24 position. And it had 4 symbiont SMTs. And so here I'm showing you the no SMT control, and you can see desmosterol. And sure enough, we see that 2 of these are actually able to methylate at the C24 position. This is significant because to this point, no bacterial SMT has ever been shown to actually be functional. And we're showing you here that it can actually methylate at the C24 position. And then we have something like this symbiont number 3, which was making these really weird ones that we didn't know what they were. It's making quality sterol, which is adding a methyl group, and then this 24-methyl desmosterol. So they're methylating, but they look different than what we were expecting. And then you have this 4th one that had no activity. So within this sponge, we had 4 symbiotic SMTs that were doing, 3 different things. The next symbiont we tested was this original one that we had found from Aplycina aerophoba. And the first one you can see, so again, just to orient you, no SMT, desmosterol. And what we see here is this one had no activity. This second one, though, completely threw us for a loop, because what we see here is we see a significant amount of methylation. We see ethylation, and then what looked like it was propylation. And this is one protein. So this is a bacterial protein on its own doing this. And so we were a little bit skeptical at first when we did this. And we were kind of afraid to say that this was actually what we thought we were saying. So Mallory went ahead and redid the experiment, but she used this molecule as a substrate. Again, because what you could have here, if you have one protein working on 2 different substrates or 3 different substrates, you're going to have competition. So if we kind of got rid of the desmosterol, could we see more? And sure enough, we do see a little bit more of the 24 isopropyl being produced when we give it this substrate instead of desmosterol. We still weren't 100% convinced. The chromatograms look good, but we weren't too sure. And so we reached out to our collaborator, Jose Hiner, who is an NMR specialist, and in particular, he's very interested in the sterol methylation, and he confirmed by NMR that this is it is an isopropyl that's being produced here. So we have a bacterial symbiont that is actually able to generate the biomarker completely on it with one protein on its own. So Mallory has gone on, I'm not going to run through all the chromatograms, but she's done so much work to test all of these different bacterial SMTs, both from sponge symbionts as well as some of the free living ones. And what you can see here is that we have the one, this is the only one that we've been able to see any propylation. We do see methylation from some of the other other sponge symbionts. The free living ones, the ones that we think are microbes that are just not symbionts of any organism potentially, are also methylating and ethylating, but none of these yet have we shown are able to propylate. Okay, so a lot of work being done here by Mallory. So what does this mean? So what I think this means is that there's actually multiple pathways to be able to propylate at the C24 position. And we could have this sponge pathway shown here, which requires, a promiscuous SMT. You can have the algal pathway, which has 3 specific SMTs to do this. And then there could be this completely different bacterial pathway where they can propylate all on their own with one enzyme. And so this is really exciting for us, because what this allows us to do now is to actually be able to do the biochemistry and see, are there differences that we can trace in the biochemistry of these that could be resulted and tested in the methylation by studying these 3 different pathways. And so Mallory's very interested in pursuing the structure of these proteins, in particular, how different at a 3D level is the SMT from this bacterial pathway versus the ones from the sponge or from the algae. It also allows us to really kind of do some pinpointing of which amino acids are important for carrying out these reactions. And the reason that's important is if we can do site-directed immunogenesis, where say we can stop this bacterial SMT from being able to propylate, what amino acids are required to do that. Then when you look for these in the genomic databases, you can begin to put them into buckets, right? Oh, this one will propylate on its own. This one can ethylate. If we can figure that out at that very granular level, and then that's just going to inform any molecular clock analysis we do, or any other types of bioinformatics that we try to get at the evolutionary pathway. So there's a lot of work we have to do still. The system is working really well for us in the lab, but it just opens up a whole bunch of possibilities of what we can do next. And so we're really excited about being able to pursue this further to answer some of these harder questions. And one of the hardest questions that we have left to answer, and what everyone's going to ask me, is, can we still use this as a diagnostic biomarker for sponges? And I think, yeah, I mean, I think that you still can, as long as we have the caveat that there are potentially other sources, like with any biomarker that we use, right? We're trying to refine it. We're trying to make it more robust in our interpretations. And this is just one of the added information that's helping us, right? I don't know if we find that only sponge symbiont are able to actually propylate, then does it matter, right? Is it still just a biomarker for sponges and their microbial communities, right? Or if we find these free-living organisms, are they able to propylate? What does that mean? And I think it just, we just have to, with our biomarkers, like with every biomarker that we have, we just have to kind of be very rigorous about the way we interpret it and put everything, all the potential doubts that could be out there as well, point those out. But I don't think you can just flat out say this isn't a biomarker for sponges. We do know that they make this, and how they make it is important, and we're going to get there, and it's going to be a better biomarker because of the work that's being done on these. Okay, so hopefully that satisfies everybody, but probably not. But either way, it's super exciting. Okay, so I think I'm done there, and I just want to acknowledge my wonderful group that has been just really, this has been a rough time for all of us. I miss them dearly. I hardly ever get to see them. I'm going to start crying. I hardly get to see them, but I get to see them on Zoom a lot, and I can't wait till I can be in the hallways and talk to them about science. Jose Hiner, who is our fantastic collaborator, so much fun to work with him. He's so excited about all these projects, and to see him engage with my students the way he has been fantastic. Our funding, this project is primarily funded through NSF. So great, very happy, thankful, sorry, for that. And of course, Mallory, who's done all of this work here and has just been so persistent. And she knows more about SMTs and sponges than anyone else I know. And she can find any metagenome in the dark web. I have no idea how she finds these, but she finds these SMTs. And none of this would have been possible without her hard work, especially during these hard COVID times. So, and with that, I'd be happy to take any questions.
Well, thank you, Paula, for an amazing talk. In fact, I had something prepared here to sort of say thank you. Mm-hmm. One of the things that I was finding frustrating is that, I love that, people finish a talk and then you get an applause. So I found that and I was trying it today. So mm-hmm. Yeah, thank you for the talk, and obviously to Mallory as well for doing that fantastic work. And yeah, over to questions. So there are several different questions. I don't know, Paula, if you can sort of see them. I can sort of read them to you. Do you have any idea why some organisms methylate at the C24 position? What is the benefit? What does it do to the membrane, for example? Any idea?
No. I don't think we know why these are methylated. I think yeast mutants have been generated, and I think they're viable. Mallory might have to back me up on this one, but we don't know the function, and that is really something that we don't understand for a lot of these like complex sterols that make these extra methylations. We're not sure. The dinoflagellates have a C4 position methylation. We're not sure what that does. The only thing that we can think of at the very basic level is that they are modifying the membrane in some way. But that really depends on how much they're making and how much is in the membrane. I mean, it's also possible that it could be functioning as some signaling mechanism. When certain sterols that are methylated at certain positions get accumulated, they can change the transcriptional profile of the organism, but that still hasn't been shown. So it's been preserved over time, so you think it must have a very significant physiological condition. And I think now that if we can find microbes that could do it, we could maybe, I don't know how to study sponges, so I don't know how we would study that physiologically, but if we can find microbes that do it, I might be able to get at that question.
Yeah, thank you. I think, I should have mentioned that was David Naft's asking the question. So next question is Merv. So very cool research. You showed that a methylated side chain may be due to the bacterial SMT activity. Does this suggest that the triod stain with a single methylation at 27 position may be a genetic product rather than a biomarker indicative of a source?
I'd have to go. I'm sorry, reading the question. Yeah. So so that is something else we're very interested. We have looked at those C26 sterols. We currently think they are biologically produced, but we don't have any evidence for that. We've been kind of trying to see if some of these other ones could potentially be doing that. But again, it requires us to be able to get the activity in the test tube. And so we're not sure. But I always lean towards the fact that there's some biotic activity, but I also know that there's a lot of abiotic activity that occurs in preservation that might be causing these to occur. So, but we are looking. That is always on our minds when we see these chromatograms coming.
Cool. Great, thank you for that. So, there's a comment from Roger Summers saying, this is wild and great. So good work, both of you. Let's see, are there any more questions? Connor O'Neill, do the methods you've described have the potential to refine the fossil record of animals that poorly fossilized jellyfish is a jellyfish biomarker or is a physical thing?
I think that's why we use them for microbes, right? Because they don't fossilize as well. So I haven't thought about what potentially jellyfish could be making, but I don't see if we can use them for microbes why you can't use them for jellyfish. This is probably David Gold has a better answer than I do, but I would say yes, you could potentially.
Great. So there is a question from Roger. Is the chromatogram should populate? Sorry. I think it's in the. I thought it was an is, but it's an in the chromatogram. Should population, I noted a peak at longer retention time. Do you see any evidence for adding 4 carbons to C24 or elsewhere on the side chain?
So there are some peaks further down. I think those are also seen in our negative controls. So I think those might be something that is just in the E. coli I don't know what, if we're using cell-free extract. I don't know. So there are potential, but they're in our negative controls. So we haven't really looked at them that closely. But these types of experiments sometimes generate things that we're not sure what they are, which is why we have people help us identify them. But I don't think those ones further down are actually sterile molecules. They're something else.
Cool. So lots of nice sort of messages. Probably what I'll do is once we finish, I'll save the chat so you can then have a sort of careful look once we finish of sort of all the nice messages. There is another sort of question here, I think from Tom McNally. Enjoyed this talk so much. Thanks you, Paula and Mallory. If a robotic probe like Osiris Rec came back with a sample that contained evidence of population, what would the next, what would be the next step?
I don't know. Where's Osiris Rex coming from? I don't know. I would be very surprised if we found something as complex as population. It's a pretty, requires a lot of biochemistry to do that. But in terms of what the next step would be, I think we would first think it's contamination. Do we have some microbe that we contaminated with that is on there would be my first thing. But in terms of the next step is, what is the source of it? If it's real, then what is making it? And potentially it could be a microbe if we're showing that population could occur by microbes here. So I don't think it's evidence of sponges on anywhere else.
All right. There is a question, I think Gordon Love. I don't know how to pronounce that.
Aplacina. I see it.
Should produce 26 methylated sterols as the main sterols, only trace amounts of 24 IPC. actually
Yeah,
not 100%.
that gets to one of these things that we have really struggled with. We would love to get a sponge genome, a genome of a sponge that makes a significant amount of 24-IPC, and also have access to its microbiome, right? Like the sequence of so we do not have that combination. We have tons of SMT data from sponges that don't make isopropyl. We have genomic data from those. We have microbiomes from, sponges that do make isopropyls, but we don't have the genome sequence of the sponge. So Mallory has searched to try to find that one where we could have that combination. If we could find a sponge that makes isopropyl, that we have access to its genome and access to its microbiome genomes, then I think we could put something together to see if whether there's a combination there that involves the bacterial symbiont or not. So we're kind of doing this. At this point, we were just trying to see if we could even methylate C24s in a test tube in the lab, because we have so many sequences that are from just in a database that we can now synthesize and potentially use.
Great, thank you. There is another question. People will have to excuse me for my pronunciation. Sometimes I don't really know how to pronounce this name. So Sahaj, I think, what's the best method to get indigenous biomarkers from ancient sediments, especially from Precambrian? From the Precambrian, as we face lots of problems of contamination.
Yeah, this is a question for Jochen and Roger.
Yes, exactly.
There's been a lot of work done. This is a huge problem in the biomarker field, right? It's like, and that's why I said our oldest ones are 1.6 billion because of contamination issues. But I think as a community, I think there are robust protocols that are being built about how you wash these and how you wash your samples and how you make sure you test for contamination and all the negative controls that you have to do. I think as a community has kind of agreed, but it's hard. That's one of the hard parts about biomarker research. And I think, it is what it is, and we all know that is an issue, and the community is working on it.
Great. There is another question by Florence. Interesting and great work. Do any of the bacteria that have the SMT protein synthesize sterols? Or what is the reason to produce SMT? That's a really good question. Yeah,
that's really good. And it's a hard question to answer because we don't actually have a microbe in hand, because these are all sequences from the environment or from a sponge symbiont, and we don't even know what the organisms are. So Mallory can blast the SMT or the cyclase that is next to the SMT, and it comes up as some like generic nitrospira or chloroflexus or something. So there's not even an organism in the database that's been cultured that we can, link this SMT to. So that's the first problem, is we don't know what the organisms are. Whether or not they're producing the sterols themselves, in some of them, like in that sponge symbiont, we actually found it next to a sterol cyclase, a sterol biosynthesis cluster. So we're presuming that organism that makes it actually produces sterols de novo. But we don't have the organism to show, right? And we've tested some of those other proteins in our E. coli system, and we can take oxidosqualene and give it that cyclase and make sterol. So they're functional. So we think they are making the sterols. And like I said, that would be really great if we could get that microbe, because then we have an organism that is actually methylating, and we can see why it's methylating and try to get at the physiology of it. Otherwise, I don't know why it makes SMTs. I don't know why, unless it's for the sponge or for the host. The host needs it to be methylated, and so it's doing it for the host, and it's why the host retains it and why it stays inside the host, right? And so the symbiotic relationship could be there. But I don't know if that's the main reason it would retain a microbe. But
I mean, it would be really good to sort of look at the whole sort of the symbiotic relationship and study that to kind of help complement. If there is here, a question by my student, do you have any plans to sequence a sponge and its microbiome? Is
no.
That seems that's another whole project.
Yes. No, it's hard to get, as David Gold will tell you, it is hard to get sponge samples, and it's hard to then sequence. I mean, eukaryotic sequencing is hard. So sequencing a sponge genome is a whole different beast than sequencing bacterial genomes.
Yeah, exactly. I know. It's because I'm used to sort of sequencing bacterial genome, the cyanobacteria genomes, and the idea of a eukaryote genome seems complicated.
Yeah, it's big and it's got weird things like introns.
Yeah, exactly. Yes, too complicated. I'm sure like the stuff that you do isn't. That is like really complicated. So I think Roger has, another question. Are the SMTs in the Bacterial genome? Are there any?
So the Bacterial is one of the symbionts that was present in sponges that was sequenced. Like they pulled out the genome of this microbiome. It's a bacterium, and several years ago they found like cyclase and SMT in there, right? And then they cleaned up the genome sequence, and it turned out it wasn't in there. So no, the SMTs are not in the Bacterial genome. That was a contamination before. So we don't know what microbes these are. But we have so many more sequences now that we don't even know what the organism is that is carrying the SMT.
Good. Well, I have a comment to make in terms of, I sort of you had the pie chart with the different type of microbes that are part of the sponge microbiome, and some of them are cyanobacteria. And from my molecular clock analysis, what I've sort of seen is like those cyanobacteria are the symbiotic ones form a monophyletic group, and they are sister to the group of Synechococcus and Procolococcus that evolve at the end of the Precambrian, and the divergent times sort of is really consistent with with the sponges, biomarkers, and so on. So that is sort of like a sort of independent way of looking at things. So I this is why when I saw the title of your talk, I was so keen. Any other question? I think that we've sort of rinsed Paula with lots of questions. So I don't know if anyone else. I think that's pretty much. No more questions. I don't know if anyone wants to make any comments. Oh, thank you for your answer. And can you? Oh, this is.
I can reach out to Sajid. I'll send him Joachim's number.
Yeah, maybe we should have him, in one of the talks. So I think, thank you. But this is Ahad again. Thank you for the answer. And can you please suggest any good analytical method to overcome this problem? As there's some previously applied methods.
Yeah.
Contamination during sampling. This is. I don't know how you feel about that, because
more of an analytical questions on how those are going. Yeah.
But these methods are not that powerful, unfortunately.
Yeah.
So I don't know. I think that.
Yeah, I don't know. I mean, I think it's the field is working on it. We don't work on that, but I think it's just kind of, it is one of the challenges that we have with the biomarker is the contamination, and how we can really kind of, get at this without destroying our samples and being confident that things are there. Yeah.
Very interesting. Thank you for the talk. To what extent could we rely on this method to detect biosignatures, to detect extraterrestrial life? What are the limitations? Could it be used for another kinds of life?
Yeah, I think the biomarkers we look at are a lot more complicated than what we would see on another planet at this point, in terms of what can be preserved and what life would be making on another planet. There are the limitations in our approach, and what we do is because we're looking at biochemistry that we're relying on a lot of what we know about on Earth, right? And so if you're looking at other life, we're looking at it through the prism of how life evolved on our planet, which is, I think, reasonable, because this is the only instance we know where life has evolved. So this is kind of like where we start, right? But I think the limitation of that is that we then kind of just close ourselves off to this being the only possibility, that this is like this biochemistry that we study, these proteins we study, these lipids we study, that's what we are looking for. But I think when you go beyond our planet, you have to think beyond that and be a little more like thinking that other possibilities of life could be possible, other possibilities of doing biochemistry could be possible. So I think that's the limitation of our work. But But there's so much we don't know here on our own planet that I don't mind focusing on this.
Good. Well, fantastic.